Welcome back to Dissect the Question. You administer a drug to a person doing intense leg extensor exercises. The drug inhibits creatine kinase and blocks the creatine kinase reaction. Which of the following molecular changes explains the effect of creatine kinase inhibition on this person's exercising muscles? So, where do we start? Creatine and kinase. And here in the lead-in, we have creatine kinase, but we have inhibition. So to understand this, we have to know that if we block creatine kinase, it's going to affect something here, A, B, C, D, or E, that's going to affect the contractions. So creatine is what? It's an amino acid that binds phosphate, and so we have this enzyme called creatine phosphokinase, or sometimes we just call it creatine kinase. And a kinase phosphates, doesn't it? Phosphorylates, I should say. Kinase phosphorylates something. So what are we phosphorylating during the creatine phosphokinase or creatine kinase reaction? We're phosphorylating ADP to create ATP. That's the function. And then when we use up the ATP, it gets recycled back to ADP. So creatine phosphokinase or creatine kinase controls the rate at which ATP is formed after it gets depleted by muscle contraction. Let's see if something jumps out at us as being the most likely answer based on these words that we found were clues. Well, creatine kinase, creatine phosphate, it looks like D with phosphate accumulation and E with phosphocreatine depletion would be the keys. We're not sure yet, but let's look at those first before we look at the others. Let's look at D. Phosphate accumulation strengthens the muscle contractions. So if we accumulate phosphate, phosphate is called inorganic phosphate, right? That's what we call inorganic phosphate. It's the inorganic phosphate that has to combine with ADP to make ATP. So if we allow phosphate to accumulate, okay, it's going to cause this reaction to go to ATP, isn't it? More phosphate, it's a substrate for ADP, so we'll get more ATP and it will strengthen the muscle contraction, okay? But is that what creatine kinase is doing? Okay? Let's look at phosphocreatine depletion in E. Phosphocreatine, okay? Phosphocreatine is the source of the phosphate. It's called CRP. CRP provides the phosphate, the inorganic phosphate, for this reaction to occur. So phosphocreatine depletion, weakening the muscle contractions, okay? It seems like the kinase is going to take the phosphate. So phosphate is going to stimulate the ATP. Anything else that we can look at? Creatine accumulation strengthens the muscle contractions. So if we create more creatine, it's going to bind more creatine phosphate. It's going to bind more phosphate to produce more creatine phosphate. And creatine accumulation could strengthen the muscle contraction. What about ATP depletion weakening the muscle contraction? it's clear that ATP depletion, ATP is the battery juice for the cell. It could be that ATP depletion is going to weaken the muscle contraction. But is that a function necessarily of creatine phosphate? If we deplete the ATP, we could be using it up because of the intense exercise. It would weaken the muscle contraction, but it wouldn't be a direct result of the CPK or CK reaction. So if we inhibit creatine kinase, out of those four choices, which were all very reasonable, it seems like it's the phosphocreatine depletion weakening the muscle contraction, because we don't expect creatine kinase inhibition when there's phosphate accumulation. When there's more substrate, we would get more CPK activity, wouldn't we? To make more ATP. So it seems like at this point, E is the best choice. Creatine accumulation, C, would cause there to be a lot of creatine phosphate, just like phosphate accumulation. Both of those, C and D, would strengthen the muscle contraction. That's true, but that wouldn't necessarily increase, or actually that would not meet the standard for this question, because we're looking for something that's going to inhibit creatine kinase. We're looking for something that's going to be the result of inhibiting creatine kinase, which is going to be a weakening of the muscle contraction. So while C and D could be correct concepts, strengthening the muscle contraction is not what's going to happen. So we have B or E, and it looks like E is the best one out of those two. Now again, because a multiple choice question could have more than one answer, we can't just move on without looking at A. A refers to ammonia depletion. So where does ammonia come from in muscle contraction? Well, it turns out when the muscle contraction is intense, like during intense exercise, over a long period of time, intense contraction stimulates a reaction called the aminohydrolase reaction in muscles. Aminohydrolase. Now, aminohydrolase breaks down AMP, adenosine monophosphate, to create more phosphates, more inorganic phosphate for this reaction. And that helps buffer or replenish the ATP, aminohydrolase. As a result of an aminohydrolase reaction, we get a buildup of ammonia, NH3. Amino, where does amino come from? From ammonia, from NH2, which is part of adenosine monophosphate, adenosine. Okay? And so, NH3 builds up under intense exercise. So ammonia depletion normalizes the pH of the sarcoplasm. Let's see how that works. That might be true because ammonia depletion, ammonia produces an alkaline environment inside the cell, and ammonia depletion would then produce more acidic conditions. Well, we know during intense contractions, the muscle creates more carbon dioxide, doesn't it? Which makes it more acidic. So depleting the ammonia would actually make it even more acidic, and that would not be normalizing the pH. So even though that is a wrong concept, we have to look at it in terms of learning, and we have to look at it in terms of eliminating that as a choice. So it doesn't normalize the pH of the sarcoplasm, and secondly, it doesn't have a lot to do with directly affecting creatine phosphokinase. So let's stick with E as the best option.